How up to date do you consider yourself with the modern world? Let's find it out. Here's the question. What are the most innovative ways scientists are repairing or replacing damaged tissues in the human body? Well, if your answer didn't include lab-grown organs, then you might be missing out on one of the most exciting advancements in regenerative medicine. Lab-grown organs are reshaping the future of healthcare, offering hope for patients waiting on transplants and opening doors to entirely new possibilities. Lab-grown organs are created by cultivating human cells in a controlled environment. These cells are grown on scaffolds, which provide a structure for the tissue to form and develop. Over time, the cells multiply and differentiate into functional tissues that mimic the structure and function of real organs. Scientists are able to take stem cells, which have the unique ability to become any type of cell, and guide them into becoming the specific cells needed for an organ, such as heart, kidney, liver cells, or brain organoids. Once the cells are ready, they are placed on a scaffold, 3D structure that supports the growing cells, allowing them to form layers and grow into a full organ. These scaffolds can be made from biodegradable materials, which gradually dissolve as the organ matures, leaving behind a functional living organ. While lab-grown organs offer immense promise, there are still several challenges that scientists must overcome. One of the major obstacles is vascularization, creating blood vessels within the organ so that it can receive the nutrients and oxygen needed to survive. Without these blood vessels, even the most perfectly engineered organs would fail due to lack of blood supply. To understand this process better, I conducted an experiment to stimulate oxygen delivery to lab-grown tissues. My goal was to compare how oxygen delivery would work with and without blood channels in a tissue being grown in a lab, and analyze how the material the scaffold is made from affects this process. So let me walk you through what I did. I used gelatin as the scaffold and the colored liquid as the simulated oxygen delivery. In the first experiment, after I solidified the gelatin properly, I injected the colored liquid directly into the scaffold, and as you see, injecting did not go that successfully. The liquid pulled out from the sides, the needle of the syringe went in, so I needed to use paper tissue to absorb it. The amount of red liquid that stayed inside the scaffold was very low, and even it's barely seen in the pictures. Because the scaffold was solid and the molecules of it were arranged in fixed positions, the liquid could not diffuse so much. This shows that our oxygen was not delivered to every cell. In the second experiment, I modified the scaffold by adding channels to stimulate blood vessels. These channels were created by inserting straws into the gelatin. I then injected the color liquid into these channels, mimicking the way oxygen is delivered through blood vessels in the human body. This time, the liquid spread more effectively as the channels allowed for better distribution throughout the scaffold. The liquid diffused more evenly, reaching a greater area of the tissue, which suggests that the presence of blood vessels represented by the channels improves oxygen delivery to cells within the scaffold. And one of the reasons is that blood vessels create a larger surface area for oxygen to diffuse, and they prevent uncontrollable movement of blood components, including oxygen and nutrients. For the next experiment, before solidifying the gelatin, I added vinegar instead of water. The vinegar's acidity altered the properties of gelatin, making the scaffold more flexible and less firm than the previous one. Initially, the liquid started to spread more rapidly, but after some time, the movement eventually stopped. Then I did some quick research on it before I moved on, and I found out that the rate of diffusion was faster at first because the scaffold was less firm, but it led to uncontrollable movement of the liquid. And I wanted to check the firmness of the scaffold made from vinegar, so I removed it from its container. It was very soft, almost breaking under the slightest pressure. It did actually break after I pressed once or twice. Then I prepared another new scaffold using water and checked its firmness also. The one made from water was much more turgid and resistant, and it had no signs of being about to break, even after I pressed it. For the next experiment, I prepared a solution using baking soda and used it to create the scaffold. The scaffold turned out cloudy in color, and the alkaline environment introduced by the baking soda made the structure firmer. This also made creating channels with a stroke easier. However, the liquid did not spread much, or perhaps not at all. From the side view, I couldn't observe any change due to the cloudy color of the scaffold, but when I looked from the above, even after some time, there were no visible changes, leading me to conclude that the liquid did not diffuse effectively. For the last experiment, I used a sugar solution to prepare the scaffold. The sugar created a more viscous, sticky texture compared to the previous scaffolds. This added viscosity made it more challenging for the liquid to move through the scaffold. The liquid spread very slowly, and it was difficult to see any significant diffusion. In conclusion, these experiments highlight key factors that must be considered when designing scaffolds for lab-grown organs: material properties, structure, and ability to simulate vascular channels. The gelatin scaffold, without modifications, was too rigid for effective diffusion, but adding channels to mimic blood cells improved the spread of liquid. Modifications like vinegar, baking soda, and sugar each impacted the scaffold's property from softness and instability to increased firmness and viscosity. Creating the optimum scaffold is one of the keys that bring us closer to a future where functional lab-grown organs can be reliably created and used in medical applications. Thanks for watching.